Hey folks, there's still a few minutes before I think we're supposed to get started at 8. So right now I have 7:56, which means I am going to sit here and delightedly read one of the books from the shelf of the Google AI library, which I am very fortunate to be kind of available to use this morning. And also kind of delighted, like there are a number of like little alien figurines on the walls, and also like a bunch of Play-Doh in weird shapes that I do not remember from my childhood. So this is fun times. Excited to be here with you today and to talk a little bit more about TensorFlow API documentation. And in the meantime, I'm going to grab the book and I'm going to read it aloud to you. So let's see. This is perfect. The Feynman Lectures on Physics. It's one of my favorites, actually. So let's start. The relation of physics to other sciences. Physics is the most fundamental and all-inclusive of the sciences and has a profound effect on all scientific development. In fact, physics is the present-day equivalent of what used to be called natural philosophy, from which most of our modern sciences arose. Students of many fields find themselves studying physics because of the basic role it plays in all phenomena. In this chapter, we'll try to explain what the fundamental problems in the other sciences are, but of course it's impossible in so small a space, really, to deal with the complex, subtle, beautiful matters in all these other fields. Lack of space also prevents our discussing the relation of physics to engineering, industry, society, and war, or even the most remarkable relationship between mathematics and physics. Mathematics is not a science from our point of view, in the sense that it is not a natural science. The test of its validity is not an experiment. We must, incidentally, make it clear from the beginning that if a thing is not a science, it is not necessarily bad. For example, love is not a science. So if something is said not to be a science, it does not mean that there is something wrong with it. It just means that it is not a science. 3.2, chemistry. The science which is perhaps the most deeply affected by physics is chemistry. I would challenge that. But maybe earth sciences wasn't a thing back in whenever he wrote this. Historically, the days of chemistry dealt almost entirely with what we now call inorganic chemistry, the chemistry of substances which are not associated with living things. Considerable analysis was required to discover the existence of the many elements and their relationships, how they make the various relatively simple compounds found in rocks, earth, etc. This early chemistry was very important for physics. The interaction between the two sciences was very great because the theory of atoms was substantiated to a large extent by experiments in chemistry. The theory of chemistry, i.e. the reactions themselves, was summarized to a large extent in the periodic chart of Mendeleev, which brings out many strange relationships among the various elements. And it was the collection of rules as to which substances combined with which and how that constituted inorganic chemistry. This is beautiful. I love Feynman. Oh man, he's such a cool guy. If you guys ever get the chance, like there's a phenomenal interview where he's playing bongo drums, but which is one of his many talents. He was also exceptionally good at cracking safes. So lots multi-talented Mr. Feynman in lectures on physics. I actually kind of like that. I will read a short snippet of lectures, the Feynman lectures, before every session I do from now on. But regardless, given that, let's get started with talking a little bit about TensorFlow documentation. So if you have dealt with the API docs in any form or fashion since using TensorFlow, you've probably noticed that some of them are a little bit more cryptic than others. Some have links to files that can be readily associated with each method or each endpoint. And it's kind of straightforward as to where you would go about making changes in order to have those reflected in the documentation. But others are a little bit more cryptic. And those are in particular the ones I want to talk about today. So the first thing we're going to do is we're going to take a look at one of the endpoints that can be modified with a Python file. Second thing, we're going to take a look at one of the math operations that's defined in like this shady .cc file where you have to modify a .doc. And then the third one is one that I didn't even know about until like 2 a.m. last night. Well, maybe a little bit before 2 a.m., but it was still ridiculously early. A collection of .pb text files. APB, my initials. PB text files that actually define some of the documentation that we have for a large number of endpoints. And they have this wonky syntax as well that takes a little bit of getting used to. But the good news is that I will hold your hand, we will walk through all of this, and we will come out shining on the other side. So let's get started. Excellent. I'm going to go ahead and share my screen. Cool. So what we have here is tf.module, and tf.module is going to be really extremely important in TensorFlow 2.0. So if you haven't taken a look at its documentation, I strongly suggest that you do. You can see here that it has a lot of sort of dialogue around what it is. It's a base neural network class. It has usage examples with sort of this well-articulated rationale for using it, including with TF function. But if you notice, like these usage examples, they're a little bit wonky. It's obviously Python syntax. But for whatever reason, they aren't formatted correctly on the page, and that's because these .py files use the typical Markdown format that you would see for making any modification on GitHub. So the backtick, 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 and then whatever language would happen to be, and then you close it out with another backtick, backtick, backtick. So given that, let's go and take a look at this linked .py file that we have here, this Python module.py. And it would probably behoove me to mention, if you bounce over to the TensorFlow 2.0 API docs FAQ, you should have a clear description of what would constitute a good usage example, what would constitute a good alias, parameters defined, returns defined. And there's also a link to the documentation tracker, which shows you that tf.module has not yet been defined, but it would be here. So let's get started. So here we have the TF export for module. We have it listed as a class, and we can check to make sure that this is the same sort of verbiage that we see here. So base neural network module class, base neural network module class, and the doc string is what dictates what happens in the documentation. We can also see that GitHub was giving pointers that this was probably not correct syntactically for being included in this doc string, but we should go in and change it regardless. So that's line 173. One of the nice things about GitHub is that if you want to make corrections just sort of inline in your web browser, you can click edit this file, just the little pencil mark. You can scroll all the way down, So you can see here, here's the TF export module. And then you can just add the backtick, Python, backtick. And it should be formatted in the way that it would be. Let's do this again with this guy. So backtick Python, backtick should be this guy as well. Backtick Python. There you go. And then this last one. So talking about variables. And close it up. And that should make all of our usage examples formatted appropriately with Python. Format usage examples in docstring for Python. Make it so that everybody knows it's my Google account. Commit changes. And create a pull request. And it'll go through a series of checks. You can see that it has to be approved by a number of folks before it gets added officially. And I'm going to go ahead and assign some tags so that people know it's related to docs and related to 2.0. Cool. Cool. And that's really as simple as it. That's really as simple as the modification of a Python file would be. Is you notice something looks amiss in the documentation, that this maybe isn't what a Python terminal would delightfully accept. You click the little edit button, you go and you make the modification, and you submit a pull request, and hopefully it goes through. And if it's a small scale modification like that guy, usually it goes through very, very quickly. Cool. So that was tf.module. What about something that's a little bit different, right? So if I wanted to look at this tf.math.add, adding things seems like it should be pretty straightforward, right? Like it's, you know, that seems like something that somebody might want to do. So tf.math.add, we have some aliases that are associated with that endpoint. So tf ragged tensor add, tf math.add. We have the required inputs for it. So if I bounced over to a collab where I've imported TensorFlow, I should be able to do like tf.math.add and like 2, 3. And it gives me back a return tensor, and it gives me the NumPy value of it. Yeah, so 5. And that's cool. So everything seems to work. But the description is a little bit strange, right? So it says it's defined in a generated file, this genmathopspy, whatever that happens to be. And if you bounce over to GitHub, you will be very perplexed to find that this file does not in fact exist. It says that it returns x plus y element-wise. You would think that this would be much better formatted with a backtick, right? Maybe it would be useful to have a usage example. And it also has a note. Math.add, which is correctly formatted, supports broadcasting. Addn does not. More about broadcasting here, and it's also missing a period. And that looks like it's going to syfy, which at least is the correct link, right? But it's also like addn. That doesn't seem like it's probably referring to this guy, which is now add_n, which does not support broadcasting. So at least that's correct. But it looks like, okay, so number of things that we should change here. We should change x plus y to be correctly formatted. we should change addn to be tf.math.add_n, and we should probably add a period to this syfy link, even though it's a little bit wonky in the first place to have a link to other other documentation sources that potentially don't potentially might change their URL links, and then we would all be very upset and very sad. Cool. So, fun fact, these guys, if you wanted to modify the docstring for this, you would have to go to this file, which is located in TensorFlow core ops, mathops.cc, and change the docstring. So if you do a control F.doc and bounce down, you can see that this is what is generating the doc for tf.math.add. So returns x plus y element-wise, returns x plus y element-wise, adds supporting broadcasting, addn does not, and then more about broadcasting there with the missing period. So that's line 397. Let's go in and make those changes that I just mentioned, and then we'll have a discussion about why this is kind of strange to see, and also kind of not exactly the what you would want a hunt down. So reformatted x plus y and changed tf.math.add and tf.math.add_n, more about broadcasting here, and add a period. So reformatted an example. And actually, maybe we should include a usage example as well, right? That seems like it might be a good. Well, let's just do one thing at a time then. So reformatted an example. Reformatted example variables. Fixed typo. And commit changes. Create pull request. And you can see in this .doc, this .dot example, that it accepted sort of the same sort of formatting that you would have in a Python docstring. If you skipped a line, the line was skipped. If you used backticks, the backticks kind of preserved the functionality that you wanted. But it's a little bit strange to think that you would have to go and find all of those sort of wonky, it's not like, it feels a little bit like you're on a quest to find a lamp in some dark, deep cave, right? Like what kind of magic is associated with being able to understand where all of these nuanced locations would be for documentation examples. And this is going to sound terrible, but one of the best things that you could do is to find where, if you have, so for this one, at least you have a file that you can go and point to. For argmin, you've got a file that you can point to. For arcsine, it's another genmathopspy. So if you wanted to find where this would be in the TensorFlow documentation, just look for the one sentence that does not look like it was generated. So computes a sign of x element-wise, which is also a terrible just description of a method, but whatever. Look for it, put it in quotation marks in this repository, and it takes you to the location. So you'll see computes a sign of x element-wise. And that leads us to our next example, which is the delightful reality that there are a number of sort of base API method descriptions and usage examples embedded within TensorFlow Core, API Def, Base API. All of these PB text files. You can see everything from audio summary to barrier close, to Bessel to boosted trees, broadcast gradients, hundreds and hundreds of these examples, some of which are exposed as actual endpoints and others which aren't. So for example, let's take a look at this guy. You can see that there's an op, a graph op name, some in arguments, some out arguments, a summary, a description, and this is actually a fairly decent one, right? So it goes through and it's correctly formatted. It has some wonky syntax with ends and like the sort of the less than sign ends, but it's at least something that you can point to in order to make a change. So let's take a look at this arc cosine example, or arc sine example rather, sorry. All right, so I want to find, I want to create a usage example, and you must be on a branch to make or propose changes to this file. Cool. So let's master change, and let's just change that. X should also have a sign. F.math.asign of x element-wise. So that's a summary. We've got the graph op name. Then I would add description. And I would want to have a usage example. So,
oh, sorry. I'm
sorry, no worries. A usage example. For example, if I wanted to compute the arc sine of, let's see, tf.math.arcsine. Let's bounce over here. Let's see. And it has to be a tensor. So let's see. Wonder if let me think of what a good example would be. Or if I didn't want to have to think of what a decent example would be, what I could do is go to the syfy documentation, right? So a sign of syfy. Isn't it fun whenever the methods are called different things? It goes through and it has notes. So arcsine is a multi-valued function. For each x, there are infinitely many numbers c such that sine of z equals x. And then the examples that it gives, kind of like this. So it does give just kind of a basic 1, a -1, and a 0. Interesting. So I wonder if I was going to do this for... Oh, gross. That's a terrible error message. But it does tell me that I don't have, that I'm not using a tensor, that I'm not using a tensor as my input argument. So what I could do is just create a tensor with one value. It could be 1, -1, and 0. Make that modification, and then the PB text file would have what it needed in order to have a clear usage example. But that would take additional time. And we're already kind of running close to the 30-minute mark, which was when I promised to start answering your questions, as opposed to just waxing philosophic about the 3 reformatted variable names. Waxed philosophic about the different locations of TensorFlow API documentation. The good news, or additional good news, is that we also have a variety of other ways to contribute. So for example, TensorFlow examples was recently created as a place where community can contribute their Jupyter notebooks, their sort of TensorFlow Lite apps, if you would like to, and markdown files are markdown files for any TensorFlow examples that you think would be interesting to see. You can also see some course content for Udacity listed there. So if you bounce into the English version, you can look at neural style transfer. You can take a look at this example notebook that was recently submitted by Kiran. That's really cool. Wait, that is totally Eminem. Rad. Okay, so you can see very creative usage examples and notebooks that have been submitted by the community. Strongly suggest that you take a look and submit your own work. Some of this stuff is really, really cool. Let's see what Times Square would look like when painted by Monet. That's really nifty. Perfection. And Jupyter notebooks, of course, are just .ipynb files. You can also click a button and with one click deploy this in Google's Colab and run it on a CPU, a GPU, or a TPU. Excellent. So with that, I'm going to go back to this guy, stop presenting, and I don't see any questions listed. So if there aren't any questions, if there aren't any people kind of logged in, let me make sure that I don't have any, like crazy, oh no, Twitter questions. I don't see anything there either. So with that, I would like to make sure that you know that there are 3 ways to modify documentation, API documentation on TensorFlow, right? So for the linked Python files, it's really straightforward. You click the URL link. It takes you precisely to the file that you need to modify. You'll see a doc string. The doc string expects the same kind of formatting that you would have in Markdown that you would be using on GitHub anyway. And it's happy path. Everybody's very excited, and you can follow the documentation style guide that we have listed on the site. If it says it was generated by genmathopspy, could be in a couple locations. So it could be that you're modifying a C file for mathops, one of the .docs that are also formatted exactly like you would expect to see for any Markdown file. Or you could have a .pp text file embedded within that other directory that has kind of a wonky syntax. It looks like a Python dictionary or JSON, kind of. And strongly suggest that if you want to have more complicated examples, taking a look at some of those pp text files that are a bit more complicated as well. Excellent. And if there aren't any questions, I promised I would be here till 9 a.m. I will be here till 9 a.m. And what I'm going to do is I am going to continue reading from Mr. Feynman's book out loud, because otherwise I don't think anybody would be very excited to see me just like chilling in a library, answering emails or like making small gardening modifications to GitHub repos. Cool. So we were talking about chemistry, right? Considerable analysis was required to discover the existence of the many elements and their relationships, how they make the various relatively simple compounds found in rocks, earth, etc. This early chemistry was very important for physics. The interaction between the 2 sciences was very great because the theory of atoms was substantiated to a large extent by experiments in chemistry. The theory of chemistry, i.e. of the reactions themselves, was summarized to a large extent in the periodic chart of Mendeleev, which brings out many strange relationships among the various elements. It was the collection of rules as to which substances combined with which and how that constituted inorganic chemistry. Inorganic chemistry was one of my favorite classes in earth science, and for good reason, because it kind of dictates the crystal shapes of rocks as they're created and as they're deformed. All these rules were ultimately explained in principle by quantum mechanics, so that theoretical chemistry is in fact physics. On the other hand, it must be emphasized that this explanation is in principle. We have already discussed the difference between knowing the rules of the game of chess and being able to play. So it is that we may know the rules, but we cannot play very well. It turns out to be very difficult to predict precisely what will happen in a given chemical reaction. Nevertheless, the deepest part of theoretical chemistry must end up in quantum mechanics. That's why everybody loves the idea of quantum computers. There's also a branch of physics and chemistry which was developed by both sciences together and which is extremely important. This is the method of statistics applied in a situation in which there are mechanical laws, which is aptly called statistical mechanics. In any chemical situation, a large number of atoms are involved, and we have seen that the atoms are all jiggling around in a very random and complicated way. If we could analyze each collision and be able to follow in detail the motion of each molecule, we might hope to figure out what would happen. But the many numbers needed to keep track of all of these molecules exceeds so enormously the capacity of any computer, and certainly the capacity of the mind, that it was important to develop a method for dealing with such complicated situations. Statistical mechanics, then, is the science of the phenomena of heat, or thermodynamics. Inorganic chemistry is, as a science, now reduced essentially to what are called physical chemistry and quantum chemistry. Physical chemistry to study the rates at which reactions occur and what is happening in detail. how do molecules hit, which pieces fly off first, etc., and quantum chemistry to help us understand what happens in terms of the physical laws. The other branch of chemistry is organic chemistry. Less cool. I didn't like Orgo. I didn't like Orgo at all. Like, the only people I know who like Orgo are the people who like memorizing things. I really did not like Orgo. Organic chemistry, the chemistry of the substances that are associated with living things. If you liked Orgo, I'm sorry. For a time, it was believed that the substances which are associated with living things were so marvelous that they could not be made by hand from inorganic materials. This is not at all true. They're just the same as the substances made in inorganic chemistry, but more complicated arrangements of atoms are involved. Organic chemistry obviously has a very close relationship to the biology which supplies its substances, and to industry, and furthermore, much physical chemistry and quantum mechanics can be applied to organic as well as to inorganic compounds. However, the main problems of organic chemistry are not in these aspects, but rather in the analysis and synthesis of the substances which are formed in biological systems and living things. This leads imperceptibly in steps towards biochemistry, and then into biology itself, or to molecular biology. Biology. Thus, we come to the science of biology, which is the study of living things. In the early days of biology, the biologists had to deal with the purely descriptive problem of finding out what living things were, what living things there were. And so they just had to count such things as the hairs of the limbs of fleas. After these matters were worked out with a great deal of interest, the biologists went into the machinery inside the living bodies. First, from a gross standpoint, naturally, because it takes some effort to get into the finer details. There was an interesting early relationship between physics and biology, in which biology helped physics in the discovery of the conservation of energy, which was first demonstrated by Mayer in connection with the amount of heat taken in and given out by a living creature. If we look at the processes of biology of living animals more closely, we see many physical phenomena, the circulation of blood, pumps, pressure, etc. There are nerves. We know what is happening when we step on a sharp stone, and that somehow or another, all of the information goes from the leg up. It is interesting how that happens. In their study of the nerves, the biologists had to come to the conclusion that nerves are very fine tubes with a complex wall that is very thin. Through this wall, the cell pumps ions, so that there are positive ions on the outside and negative ions on the inside, like a capacitor. Now, this membrane has an interesting property. If it discharges in one place, i.e., if some of the ions were able to move through one place, so the electric voltage is reduced there, that electrical influence makes itself felt on the ions in the neighborhood, and it affects the membrane in such a way that it lets the ions through at a neighboring point also. This in turn affects it farther along, etc., and so there is a wave of penetrability in the membrane which runs down the fiber when it is excited at one end by stepping on the sharp stone. This wave is somewhat analogous to a long sequence of vertical dominoes. If the end one is pushed over, that one pushes the next, etc. Of course, this will transmit only one message unless the dominoes are set up again. And similarly, in the nerve cell, there are processes which pump the ions slowly out again to get the nerve ready for the next impulse. So it is that we know what we are doing, or at least where we are. Of course, the electrical effects associated with this nerve impulse can be picked up with electrical instruments. And because there are electrical effects, obviously the physics of electrical effects has had a great deal of influence on understanding the phenomenon. The opposite effect is that from somewhere in the brain, a message is sent out along a nerve. What happens at the end of the nerve? There, the nerve branches out into fine little things connected to a structure near a muscle called an end plate. For reasons which are not exactly understood, when the impulse reaches the end of the nerve, little packets of a chemical called acetylcholine, that's such a cool name, acetylcholine, are shot off 5 or 10 molecules at a time, and they affect the muscle fiber and make it contract. How simple. What makes a muscle contract? A muscle is a very large number of fibers close together containing two different substances, myosin and actomyosin. But the machinery by which the chemical reaction induced by acetylcholine can modify the dimensions of the molecule is not yet known. Thus, the fundamental processes in the muscle that make mechanical motions are not known. I wonder if that's still true. Biology is such an enormously wide field. There are hosts of other problems that we cannot mention at all. Problems on how vision works, what the light does in the eye, or how hearing works, etc. The way in which thinking works, we shall discuss later under psychology. Now, these things concerning biology, which we have just discussed, are from a biological standpoint really not fundamental at the bottom of life, in the sense that even if we understood them, we still would not understand life itself. To illustrate, the men who study nerves feel their work is very important, because after all, you cannot have animals without nerves, but you can have life without nerves. Plants have neither nerves nor muscles, but they're working and they're alive just the same. So for the fundamental problems of biology, we must look deeper. When we do, we discover that all living things have a great many characteristics in common. The most common feature is that they're made of cells, within each of which is complex machinery for doing things chemically. In plant cells, for example, there is machinery for picking up light and generating sucrose, which is consumed in the dark to keep the plant alive. When the plant is eaten, the sucrose itself generates in the animal a series of chemical reactions very closely related to photosynthesis, and its opposite effect in the dark in plants. In the cells of living systems, there are many elaborate chemical reactions, in which one compound is changed into another and another. To give some impression of the enormous efforts that have gone into the study of biochemistry, the chart in figure 3.1 summarizes our knowledge to date on just one small part of the many series of reactions which recur in cells, perhaps 1% or so of it. Here we see a whole series of molecules which change from one to another in a series or cycle, in a sequence or cycle of rather small steps. It is called the Krebs cycle, the respiratory cycle. Each of the chemicals in each of the steps is fairly simple in terms of what change is made in the molecule. But, and this is a centrally important discovery in biochemistry, these changes are relatively difficult to accomplish in a laboratory. If we have one substance and another very similar substance, the one does not just turn into the other because the two forms are usually separated by an energy barrier or hill. And because everybody should see the pretty picture. Woo. Cool. There was a show when I was growing up called The Adventures of Pete and Pete, and it was amazing. And there was a Krebs company that worked there, or that was featured in the show. Consider this analogy. If we wanted to take an object from one place to another at the same level but on the other side of the hill, we could push it over the top, but to do so requires the addition of some energy. Thus, most chemical reactions do not occur because there is what is called an activation energy in the way. In order to add an extra atom to our chemical requires that we get it close enough that some rearrangement can occur, then it will stick. But if we cannot give it enough energy to get it close enough, it will not go to completion. It will just go partway up the hill and back down again. However, if we could literally take the molecules in our hands and push and pull the atoms around in such a way as to open a hole to let the new atom in, and then let it snap back, we would have found another way around the hill, which would not require extra energy, and the reaction would go easily. That's so sneaky. I love it. Now, there actually are in the cells very large molecules, much larger than the ones whose changes we have been describing, which in some complicated way hold the smaller molecules just right so that the reaction can occur easily. These very large and complicated things are called enzymes. They were first called ferments because they were originally discovered in the fermentation of sugar. In fact, some of the first reactions in the cycle were discovered there. I wonder why. Fermentation. In the presence of an enzyme, the reaction will go. An enzyme is made of another substance called protein. Enzymes are very big and complicated, and each one is different, each being built to control a certain special reaction. The names of the enzymes are written in figure 3-1 at each reaction. Sometimes the same enzyme may control two reactions. We emphasize that the enzymes themselves are not involved in the reaction directly. They do not change, they merely let an atom go from one place to another. Having done so, the enzyme is ready to do it to the next molecule, like a machine in a factory. Of course, there must be a supply of certain atoms and a way of disposing of other atoms. Take hydrogen, for example. There are enzymes which have special units on them, which carry the hydrogen for all chemical reactions. For example, there are 3 or 4 hydrogen-reducing enzymes, which are used all over our cycle in different places. It is interesting that the machinery which liberates some hydrogen at one place will take that hydrogen and use it somewhere else. The most important feature of the cycle of figure 3-1 is the transformation from GDP to GTP, guanidine diphosphate to guanidine triphosphate, because the one substance has much more energy in it than the other. Just as there is a box in certain enzymes for carrying hydrogen atoms around, there are special energy-carrying boxes which involve the triphosphate group. So GTP has more energy than GDP, and if the cycle is going one way, we are producing molecules which have extra energy and which can go drive some other cycle which requires energy, for example, the contraction of muscle. The muscle will not contract unless there is GTP. We can take muscle fiber, put it in water, and add GTP, and the fibers contract, changing GTP to GDP if the right enzymes are present. So the real system is in the GDP-GTP transformation. In the dark, the GTP, which has been stored up during the day, is used to run the whole cycle around the other way. An enzyme, you see, does not care in which direction the reaction goes, for if it did, it would violate one of the laws of physics. Physics is of great importance in biology and other sciences for still another reason that has to do with experimental techniques. In fact, if it were not for the great development of experimental physics, these biochemistry charts would not be known today. The reason is that the most useful tool of all for analyzing this fantastically complex system is to label the atoms which are used in the reactions. Thus, if we could introduce into the cycle some carbon dioxide which has a green mark on it, and then measure after 3 seconds where the green mark is, and again measure after 10 seconds, we could trace out the course of the reactions. What are the green marks? Well, they're different isotopes. We recall that the chemical properties of atoms are determined by the number of electrons, not by the mass of the nucleus. There can be, for example, in carbon, 6 neutrons or 7 neutrons, together with the 6 protons which all carbon nuclei have. Chemically, the 2 atoms C12 and C13 are the same, but they differ in weight, and they have different nuclear properties, and so they are distinguishable. By using these isotopes of different weights, or even radioactive isotopes like C14, which provide a more sensitive means for tracing very small quantities, it is possible to trace the reactions. Now we return to the description of enzymes and proteins. All proteins are not enzymes, but all enzymes are proteins. Okay. All proteins are not enzymes, but all enzymes are proteins. It's like that square and rectangle thing, I guess, in math. There are many proteins, such as the proteins of muscle, the structural proteins, which are, for example, in cartilage and hair, skin, etc., that are not themselves enzymes. However, proteins are a very characteristic substance of life. First of all, they make up all the enzymes, and second, they make up much of the rest of living material. Proteins have a very interesting and simple structure. They are a series or chain of different amino acids. There are 20 different amino acids, and they all can combine with each other to form chains in which the backbone is CONH and etc. Proteins are nothing but chains of various ones of these 20 amino acids. Each of the amino acids probably serves some special purpose. Some, for example, have a sulfur atom at a certain place. When two sulfur atoms are in the same protein, they form a bond. That is, they tie the chain together at 2 points and form a loop. Another has extra oxygen atoms, which make it an acidic substance. Another has a basic character. Another acidic substance. Another has a basic characteristic. Some of them have big groups hanging out to one side so that they take up a lot of space. One of the amino acids, called proline, is not really an amino acid, but an amino acid. Ah, that's adorable. There is a slight difference with the result that when proline is in the chain, there is a kink in the chain. If we wish to manufacture a particular protein, we would give these instructions. Put one of those sulfur hooks here. Next, add something to take up space, then attach something to put a kink in the chain. If in this way, we will get a complicated-looking chain hooked together and having some complex structure. This is presumably just the manner in which all the various enzymes are made. One of the great triumphs in recent times, since 1960, was at last to discover the exact spatial atomic arrangement of certain proteins, which involves some 56 or 60 amino acids in a row. Over 1,000 atoms, more nearly 2,000 if we count the hydrogen atoms, have been located in a complex pattern in two proteins. The first was hemoglobin. One of the sad aspects of this discovery is that we cannot see anything from the pattern. We do not understand why it works the way it does. Of course, that is the next problem to be attacked. Another problem is, how do the enzymes know what to be? A red-eyed fly makes a red-eyed fly baby, and so the information for the whole pattern of enzymes to make red pigment must be passed from one fly to the next. This is done by a substance in the nucleus of the cell, not a protein, called DNA, short for deoxyribose nucleic acid. This is the key substance which is passed from one cell to another. For instance, sperm cells consist mostly of DNA and carries the information as to how to make enzymes. DNA is the blueprint. What does the blueprint look like and how does it work? Well, first, the blueprint must be able to reproduce itself. Secondly, it must be able to instruct the protein. Concerning the reproduction, we might think that this proceeds like cell reproduction. Cells simply grow bigger and then divide in half. Must it be thus with DNA molecules, then, that they too grow bigger and then divide in half? Every atom certainly does not grow bigger and divide in half. No, it's impossible to reproduce a molecule except by some more clever way. The structure of the substance DNA was studied for a long time, first chemically to find the composition, and then with X-rays to find the pattern in space. The result was the following remarkable discovery. The DNA molecule is a pair of chains twisted upon each other. The backbone of each of these chains, which are analogous to the chains of protein, but chemically quite different, is a series of sugar and phosphate groups, as shown in figure 3.2. Now we see how the chain can contain instructions, for if we could split this chain down the middle, we would have a series, B, A, D, C, and every living thing would have a different series. Thus, perhaps in some way, the specific instructions for the manufacture of proteins are contained in the specific series of the DNA. Attached to each sugar along the line and linking the two chains together are certain pairs of crosslinks. However, they are not the same kind. There are 4 kinds, called adenine, thymine, cytosine, and guanine, but let us call them A, B, C, and D. The interesting thing is that only certain pairs can sit opposite each other. For example, A with B and C with D. These pairs are put on the two chains in such a way that they fit together and they have a strong energy of interaction. However, C will not fit with A, and B will not fit with C. They will only fit in pairs, A against B and C against D. Therefore, if one is C, the other must be D, etc. Whatever the letters may be in the chain, each one must have its specific complementary letter on the other chain. Cool. Excellent. So it's been an hour. I'm going to check Twitter one more time to make sure that nobody's asked any questions. Oh man. Okay, so I haven't seen Margaret's question come through, which is terrible. And I'm super glad that you asked it on Twitter. Making docs changes themselves and the examples repo. Awesome. So the example data set. I think it would probably be best to share a link to but then again, if chat isn't working, that might not be the best bet. So github.com/tensorflow/datasets should have a subdirectory called structured. So if you click TensorFlow datasets and then structured, you should see an example called Titanic.py. And let me pull this up really quickly. It was created by my colleague Yash, who is amazing. And what it does is it goes and looks at a specific CSV dataset on something called openml.org. So openml.org. And you can see here that there are a large number of data sets that folks can use, right? So 2,547. We would love to have all of them, right? So as many as folks want to add, we would be delighted to have them. And the way that you would go about ingesting these data sets is you would have a template exactly like this. So exactly like Yash's Titanic example. You would have a feature dict for each one of the features that you have associated in the CSV file. So let's take a look at this URL. Control C, Control V. Excellent. So this is a little bit wonky in terms of zoominess, but yeah. So you can see that there are names. There's sort of age, ticket, fare, cabin, embarked, boat, whatever. Each one of those features has kind of a feature type associated with it. So if you look for the example Titanic data set here, you would be able to see that all of those 14 features have already been defined. So people have mentioned as to whether they're nominal, whether they're numeric, continuous, whether they're a string value, whether they have missing values, etc. So 14 fields. And then you take a look here, and all of them are defined in this feature dict. So the only thing that you would really be responsible for is changing for a new CSV file, which features are included, what the names of those features are, and then what the type would be for each one of those features. There are also some helper functions, so convert to float, convert to ent, convert to string, convert to label. There's a description that you would want to fill in, but again, OpenML has already done kind of the heavy lifting of providing a description. So you can see here the source, the author, the description of the data set. You would fill those into the applicable fields with the citation. The description would be included here. Supervised keys indicate that that we have a column that sort of subdivides this into categories. So you have the features, and then you have a survived column, which is basically giving a 0 or 1 as to whether or not the human or whether or not the passenger survived or not. You would have split generators to divide into train and test sets. Though you should be able to just use this same one, because I don't think any of the data sets in OpenML have predefined trained validation and test splits. And then you would generate examples. So really, you just need to tweak a few things. So this feature dict, the citation and the description, and then the variable names associated for the generation of examples for the CSV files that are located there. And then there are also some tests that you can run. So here you see checking to make sure that the splits are validated and that you're able to actually ingest the thing. But that's it. So if you wanted to go hog wild and you wanted to formulate generators for all 2,400 of those data sets, you absolutely could, and all of them could get added to TensorFlow dataset structured. And in reality, the only small changes that you would have to make to that template example that Yash made was changing around the feature names and the values associated with them. So again, if just making sure that if it's continuous numeric, you label it as such. If it's a name with a string, you label it as such. If it's a sort of categorical feature, you label it as such. And then that also the citations and the descriptions are correct. And that's sort of the reality of the TensorFlow datasets. It's that easy. All you have to do is sort of define that data ingestion pipeline, and then you're able to pull in data sets at will. So does that answer your question, Margaret? I'm sorry that I didn't see it until just now. Cool. Excellent. And in that case, I am going to hope that this was useful, and also probably put all of this down in a doc because it's a little bit complicated. But again, if you want to contribute a data set to TensorFlow docs, or sorry, if you want to contribute a data set to TensorFlow datasets, and it's a structured data set from OpenML, you can probably follow Yash's template very, very closely and be able to ingest any number of the 2,400 that are currently associated there. If you want to modify documentation, there are 3 locations, as mentioned before. And if you would like to include an example, all you have to do is make a pull request and place your Jupyter notebook there. Thank you so much for listening, and have a great day.